Hey, welcome back everyone for more proteomics. So in the last few videos, we have mentioned several times that under certain circumstances, the cell may decide to destroy or degrade a protein. So in this video, we're going to discuss that very topic, how and why the cell may choose to destroy a protein. So let's go ahead and get into this. So here's the deal. So proteins, just like a machine, proteins are only going to be functional for a certain amount of time. So machines break down, appliances break down, so you're familiar with that throughout your daily life. Proteins are the same way. Proteins are really only going to be functional for a relatively short amount of time, usually somewhere in the neighborhood of hours. So the idea here is that how much protein is actually in the cell is going to be determined by kind of a balance between making new protein to replace the old ones and then degrading the old ones that are no longer functional. So how much of a particular protein is actually in the cell at any given time is going to be balanced by those two sides of it. So proteins are destroyed by enzymes that are called proteases. In an earlier video in the semester, we talked about the enzymes pepsin and trypsin and how they have different pH requirements. So pepsin and trypsin were both proteases. They are enzymes that break the peptide bonds between amino acids by using the hydrolysis reaction. And like I said, we already saw two examples of this earlier in the semester, pepsin and trypsin, which even though they have different pH requirements because they're located in different places, they do pretty much the same sort of thing. But here's the deal, not every protease is created equal. So most proteases are actually fairly specific for particular sequences or stretches of amino acids that they'll actually cleave. So let's look at a few examples here. So first are proteases called caspases. Because of the name, the C in caspases stands for cysteine and the ASP stands for aspartate. This tells us that caspases will cut in between cysteine and aspartate residues within proteins. Now, you're not going to find very many sequences that have cysteine, aspartate. You're only going to find these every now and then. So caspases appear to be fairly specific. They'll only cut between these two residues, so they're probably only going to cut very specific proteins in the cell. And as such, caspase proteases are actually fairly specific in what they do. They help to trigger apoptosis, which is the process of a cell undergoing programmed cell death. Trypsin we've talked about before. This is another protease that has some specificity. It will only cut on the C-terminal side of lysine and arginine residues, so basic residues, as long as the next residue on the C-terminal side is not proline. We've talked about how proline is a very bulky amino acid. It kind of gets in the way sometimes. So trypsin will cut on the C-terminal side of lysine and arginine as long as there's not a proline there. And then finally, another type of protease called chymotrypsin has its own specificity. It will only cut on the N-terminal side of tryptophan, phenylalanine, tyrosine, or leucine residues. Most of those, 3 out of 4, being aromatic amino acids. So far you probably have the idea that proteases are here to destroy proteins, basically clip them apart and chop them up into tiny little pieces. Now that's not always going to be the case. Proteases can exist to activate proteins too, proteins that were previously in an inactive configuration. Let's consider the example of a protein called Angiotensinogen. Just for future reference, anytime you see a protein that ends in the designator inogen or ogen, that usually means that the protein is inactive. So in this case, angiotensinogen, it is synthesized by cells in the liver as is in an inactive form. It's not really going to have any function as it currently is. So the protein will not become active until we cleave off a big portion of the protein by cutting it with a protease. So it ends up being that the smaller piece of the protein will have some activity and then the larger piece will eventually be destroyed. So the whole purpose of angiotensinogen is that it functions in the physiological regulation of blood volume and sodium and potassium content. So angiotensinogen, like I said, is secreted by the liver, and it is activated by an enzyme called renin, which is secreted by the juxtaglomerular cells of the kidney, and then another protease called angiotensin converting enzyme, which is located in the lungs. So after these two proteases clip off pieces of angiotensinogen, the final product is a very short peptide called angiotensin II. So this peptide actually has a whole bunch of activity in being able to raise your blood pressure and stimulate sodium and potassium transport in the kidneys, whereas angiotensinogen had none of that activity. So the gain in activity here is all due to amino acids being clipped off of angiotensinogen by these proteases. And this is certainly not the only example of this. Many proteins in cells in the body are secreted and synthesized in an inactive form, and they won't be activated until a little piece of them is clipped off by a protease. So here's a little bit more information on angiotensinogen that I mentioned. So you can see the angiotensinogen, which is 255 amino acids long. It's cleaved into just a 10 amino acid fragment called angiotensin 1. And then an enzyme called angiotensin converting enzyme will convert it into a peptide that is either 8, 7, or 9 amino acids long. And then that short little peptide can function as a hormone to stimulate things like raising your blood pressure and changing the function of your kidneys. Now, I don't tell you these things because I'm expecting you to remember what the physiological response is. Just keep in mind here that none of these physiological responses that we took the time to mention would be possible without protease activity in this case. That is the key deal here. Okay, so we mentioned that proteases can be used to activate proteins. What they're usually going to be for, as we have mentioned before, is that they are there to destroy proteins, chop them up into little fragments, and then we can hopefully reuse those amino acids later on. So the question here is, how does the cell determine which proteins need to be degraded and when? So there's thousands and thousands and thousands of proteins floating around inside the cell. How do we know which ones are still functional and which ones are non-functional and need to be destroyed? So the cell actually has a way of tagging proteins that need to be destroyed, kind of how you have a way of placing things inside your house that need to be thrown away. You have a garbage bag for that. So proteins that need to be destroyed are enzymatically tagged with a very small protein called ubiquitin. So this is another example of a post-translational modification. Ubiquitin can be attached onto a protein in the same way that a phosphate or an acetate or anything else could. So this tag will be specifically attached to lysine side chains on the protein that needs to be degraded. So this enzymatic attachment will go again and again and again until a protein is attached to a big chain of ubiquitins called a polyubiquitin tag. And once that has happened, the protein that has the polyubiquitin tag will be transported through the cell and into one of several different macromolecular complexes in the cell that actually have protease functions. This big structure is called a proteasome. So a proteasome, I've always kind of thought, because I actually studied proteasomes and the ubiquitin system in grad school, proteasomes you can kind of picture as just a big trash can inside the cell. That's not a bad way of kind of thinking about what it looks like. So the idea here is that if we have our protein that needs to be destroyed, then a cascade of enzymes called E1, E2, and E3 will work together to attach a big chain of ubiquitins onto the protein that needs to be destroyed. And remember, that's going to be attached to lysine side chains. And then that protein, with its polyubiquitin chain, will be recognized by other proteins in the cell, and they will deliver this protein to the proteasome the same way that you grab a trash bag full of trash and you take it out to the trash can so that it can be taken away to the dump. So the proteasome here will take this protein and start chewing it up into little pieces, so we regain our original ubiquitin molecules, and then also we'll have short little peptides that will be chewed up into constituent amino acids. So one thing that is interesting about the proteasome is that there are several different subunits here. So the proteasome, like I said, it kind of looks like a big trash can. The central core, which is here in yellow, is called the 20S core. Remember, S is the sedimentation coefficient that we talked about before. So S stands for Svedbergs. Basically, it's a measure of how much force will it actually take to get these guys to sediment in a certain amount of time when we centrifuge them. And then we have these two caps here, which are called 19S caps. The whole structure here is called the 26S proteasome. Now, what you might be seeing here is if you're doing the math in your head, you're thinking, okay, we've got a 20S core and two 19S caps. 20S plus 19S plus 19S equals 26S. Yeah, so that doesn't make a whole lot of sense, does it? You would think it would be a total of 58 Svedbergs. Well, this is a good lesson because when it comes to Svedberg math, the numbers don't add up the way that you would expect. So what this tells us is that an intact proteasome would sediment all by itself with a coefficient of 26 Svedbergs. If you separate them into their constituent units of two separate caps and a core, the core would sediment at 20 Svedbergs, and each cap would themselves sediment at 19 Svedbergs. So the math does not work out here, and that is totally expected. So it's just kind of a good lesson to learn here. Okay, so why would we want the proteasome to break down proteins inside the cell? There's actually a couple of very good reasons here. Number one, this allows for recycling of amino acids so that the cell is going to have a good stock of them to allow for the synthesis of new proteins. So if proteins are never destroyed and the cell has to continue to make new proteins, eventually we're going to run out of amino acids, right? So the cell doesn't have an infinite supply of amino acids to build proteins out of. So it's kind of like playing with Legos, considering the Lego blocks to be kind of like the amino acids. If you're building a whole big thing out of Legos and you finish building it and then you want to build something else, you probably don't have a whole lot of Legos left. So if you want to build something else, you're going to have to break down the first thing that you built in order to free up some building blocks so that you can build something else. So the cell will actually get very sick and get really stressed out if it starts running out of amino acids. So regularly destroying proteins is a good way to replenish its amino acid stores. And then secondly, a lot of proteins that have lost activity or don't function correctly, if they are not destroyed quickly, they can end up causing a lot of problems. And this especially goes for denatured proteins. Anytime the cell recognizes a protein that is either not doing its job correctly or is unable to do its job anymore, it's important to basically cull it so it can't cause all the good healthy proteins any sort of problems. So this brings us into another way that we can destroy proteins inside the cell, and this is a little bit more extreme. This process is called autophagy, which literally means self-eating. This is a cellular process by which proteins can be destroyed as well. So usually this is going to be a part of some kind of nutrient starvation or stress response process. So like I was saying before, if a cell gets stressed out because it's running out of amino acids, it may decide to activate the autophagy system so that we can just kind of start breaking down a whole bunch of things and eventually free some nutrients up. So when we talked about the ubiquitin system before, that was very targeted and very specific. So the Ubiquitin-Proteasome System can specifically target proteins that need to be destroyed while sparing all the healthy ones that do not need to be destroyed. Autophagy ends up being a much more kind of non-specific bulk process that's just going to destroy anything it finds. So you really don't want autophagy to be active all the time. And in addition, instead of just focusing on destroying proteins, autophagy can also destroy things like organelles that are targeted for autophagy. So oftentimes you'll see mitochondria inside the cell that, similar to some proteins, they just kind of lose function or get damaged over time. The cell can destroy whole organelles by autophagy as well. So regardless of whether the target is an organelle or a big protein aggregate, these targets will be delivered to lysosomes, which are also organelles inside the cell. And these organelles contain acid-based hydrolytic enzymes that are capable of destroying those target cells, or those target organelles or proteins. So you can see kind of a picture of what's going on here. In this case, we've identified several things that need to be destroyed, like mitochondria in yellow, and then like big proteins here in green and gray. So what happens here with autophagy is that everything that needs to be destroyed gets encapsulated in a membrane-bound vesicle called an autophagosome, and then the autophagosome will fuse with lysosomes, and all those hydrolytic enzymes are able to get access to what needs to be destroyed, and everything gets broken down into its constituent monomers, including amino acids, monosaccharides, nucleotides, etc. Okay, so that's going to do it for this discussion on protein degradation in the cell. So join us for our last video of this lecture, and we will talk specifically about when protein folding and protein degradation goes wrong. So we'll be talking about several different diseases that will tie together a lot of what we talked about in the previous 3 videos. So thanks as always for your attention, and I will see you next time.